Hello everyone, myself Dr. Manjula S, working as Assistant Professor and Head of the Department of Botany in Government First Grade College, Ramanagara. Today I will explain the process of double fertilization in angiospermic plants, which is prescribed by the Bangalore University in 2nd BSc 4th semester Botany syllabus. First, let me tell you the difference between fertilization and double fertilization. As we all know, fertilization is the phenomenon of fusion of male gametes with the female gamete to form a diploid zygote, which is observed in all sexually reproducing organisms, whereas the double fertilization is the phenomenon of fusion of one male gamete with the egg to form a diploid zygote and other male gamete with the two polar nuclei to form triploid primary endosperm nucleus. So as the name itself indicates, double fertilization includes 2 times fertilization. It is a characteristic feature of all angiosperms, which was first discovered by S.G. Nabashin in Lilium and Fritillaria species. Double fertilization is the complex mechanism of fertilization observed in flowering plants where there is a joining of female gametophyte with the two male gametes. In this diagram, we can observe the process of double fertilization where after the liberation of sperm through the pollen tube, one of the sperm enter into the embryo sac and it get fuses with a egg nucleus to form a diploid zygote, and another sperm fuses with the two polar nuclei to form triploid primary endosperm nucleus. So let me explain the steps involved in double fertilization. The first one is pollen germination, which is followed by growth of pollen tube through style. This step is followed by the third step called entry of pollen tube into the ovule, which is followed by entry of pollen tube into the embryo sac. Next step is called the pollen tube discharge, which is followed by syngamy and triple fusion. So let me explain each step in detail. First step, pollen germination. Before pollen germinate, there should be the phenomenon of pollination. Pollination is nothing but transfer of pollen grains from the anther of a flower to the stigmatic surface, which is carried out by two means, abiotic and biotic. Abiotic means includes wind or water. The pollination by wind is otherwise known as anemophily, whereas the pollination by water is known as hydrophily. The biotic means of pollination includes insects, birds and animals, where in case of angiospermic plants, most of the pollination is by means of biotic, that too by insects, which is called as entomophily. After the pollination, the pollen grain come and fall on the surface of the stigma, where it will start its germination. The pollen germination has 3 important steps, which are as follows. First one is called adhesion. Adhesion is nothing but the deposition of pollen grain on the stigmatic surface, which is followed by the second step, that is called hydration. Hydration is the absorption of water and nutrients by the pollen grain from the nectary present on the surface of the stigma. By this process, the pollen grain will increase in its size, and it is followed by the third step, that is called the germination of the pollen grain, where the tube cell enlarges and comes out of the pollen grain through one of the germ pore to produce a tiny pollen tube, which runs through the style till it reaches the ovary. After that, the tube nuclei descends to the tip of the pollen tube, followed by the generative cell which passes into the fallopian tube, which later divides and forms two male gametes, which are otherwise known as the sperms. In this slide, we can observe that after the pollination, pollen will come and land on the surface of the stigma, where they will start germinating by producing a germ tube, and into the germ tube the generative cell enters and it divides to form two sperm nuclei, and after that the pollen tube grow through the stylar region and it reaches the ovule. The second step is called growth of pollen tube through the stylar region. After the germination, the pollen tube must drill its way through the nutrient-rich style and curl to the bottom of the ovary to reach an ovule. So depending on the internal structure, the style of a pistil is categorized into three types. First one, hollow style. Second one, solid style. Third one, semi-solid style. Hollow styles are otherwise known as open styles, which are mostly found in monocot plants. Here there is a hollow canal which runs from the stigma to the base of the style, which is lined by a single layer of glandular canal cells. These glandular canal cells secrete mucilaginous substances which contain carbohydrates, lipids and some enzymes along with compatibility controlling proteins. These secretions serve as nutrition for the growing pollen tube. The second type of style is called the solid style, which is otherwise known as closed style, which is a characteristic features of most dicots. Here there is a transmitting tissue which is equivalent to the lining cells of hollow style. Its content are also similar to the lining cells, where the pollen tube grows through the intercellular spaces of this transmitting tissue. The third type of style is called semi-solid style, which is otherwise known as half-closed style, which is the intermediate between the solid and open style. In this diagram, we can find the difference between the hollow style and solid style, where the diagram A indicates the transverse section of hollow style present in the monocot plant Lilium, which belongs to the family Liliaceae, where there is a hollow space through which the pollen tube can easily pass through, whereas in the diagram B there is a solid mass of transmitting tissue through which the pollen should enter. So this is the difference between the hollow style, where there is a hollow space, and in the solid style there is no hollow space. Then coming to the next step, that is entry of pollen tube into the ovule. The pollen tube may enter the ovule by following three methods: porogamy, chalazogamy, mesogamy. Before telling this, let me explain the structure of mature ovule, where there are different structures like the integuments, which cover the outermost layer, which will open by means of a small opening called micropyle. So exactly opposite to the micropyle, there is a region called chalaza, and there is a nucellus followed by the embryo sac, which in turn encloses 8 nucleus. So depending upon the entry of the pollen tube into the ovule, as I already told, there are three types. If the pollen tube enters the ovule through the small opening called micropyle, then this is called the porogamy, which is observed in lily, which is the most common method in all the flowering plants. The second type is called the chalazogamy. Here the entry of the pollen tube is through the chalazel portion. It is less common and it is observed in casuarina. The third type is called mesogamy, where the pollen tube enters the ovule through its Other parts like integuments are through funicle. This is called the mesogamy, which is observed in cucurbita. Then next step is entry of pollen tube into the embryo sac. The growth of the pollen tube towards the ovary, ovule and embryo sac is due to the presence of certain chemical substances present in the ovule. Irrespective of the place of entry of pollen tube into the ovule, it may be porogamy, chelazogamy or mesogamy, the pollen tube must enter the embryo sac only at the micropylar end. The pollen tube, after traveling the whole length of style, it enters into the ovary locule where it is guided towards the micropyle of the ovule by a special structure called obturator. Obturator is an outgrowth which will guide the pollen tube to enter through the micropyle. The pollen tube enters into the embryo sac directly through one of the synergids. After reaching the embryo sac, a pore is going to be formed in the pollen tube wall at its tip. The content of the whole pollen tube is discharges into the embryo sac. In this slide, we can observe a special outgrowth at the funicle region called obturator. An obturator is an outgrowth of funicle or integument or style near the micropyle which guide the pollen tube towards the micropyle region of the ovule. So in this slide we can see the process of entry of pollen tube into the ovule, where in first step after pollination the pollen grain will come and sit on the surface of the stigma. After hydration they will start by producing a pollen tube. This pollen tube will pass through the stylar region. It will curve and it will reaches the ovule, mainly due to the chemical substance present in the ovule. After it reaches the ovule, the synergids will receive the sperm nuclei present in the pollen tube, and after the discharge of the sperm nuclei into the embryo sac, one of the sperm fuses with the egg nucleus to form a diploid zygote, whereas another sperm fuses with the two polar nuclei to form triploid primary endosperm nucleus. Then I'll come to the important process found in double fertilization, that is syngamy, triple fusion and the phenomenon of double fertilization. The male gametes, which are otherwise known as sperms, released from the male gametophyte, they are involved in fertilization of two different components of the embryo sac, where one of the male gametes fuses with the egg nucleus to form diploid zygote. This phenomenon is called syngamy, fusion of gametes, male and female gamete. This phenomenon is called syngamy, which results in the formation of diploid zygote, which later develops into an embryo. And the second male gamete migrates to the central cell, where it fuses with the two polar nuclei or their fusion product called secondary nucleus, and thus forms the primary endosperm nucleus, which later develops into an endosperm. Since this involves the fusion of three nuclei, one from the chelagial end and another nuclei from the micropylar end and one more nuclei from the sperm. Since it involves the three nuclei, this phenomenon is called triple fusion. Since both the male gametes are involved in fertilization, the phenomenon is called double fertilization, and it is unique to the angiosperms. So in this slide we can see the different stages in double fertilization. First, after the pollination, pollen grain come and fall on the stigmatic surface, where it will germinate and form the pollen tube. Then second step, entry of the pollen tube through the stylar region. The germinated pollen tube grow and enter through the stylar portion of the pistil, where there are 2 sperms are also enter into the pollen tube, and they will grow, pollen tube grow till it reaches the ovule. Once the pollen tube reaches the ovule, according to the synergid's signal, it will discharge the sperm nuclei into the embryo sac, where one of the nuclei fuses with the egg cell to form diploid zygote, and another sperm fuses with the polar nuclei to form triploid primary endosperm nucleus. In this diagram, we can observe the process of double fertilization in a very close-up view, where I told after the discharge of the sperms into the embryo sac, one of the sperm or male gamete fuses with a egg nucleus to form a diploid zygote, which later develops into an embryo, whereas the another sperm fuses with the 2 polar nuclei to form triploid primary endosperm nuclei, which is going to develop into an endosperm. Next, I will explain the development of embryo and endosperm. As I already told, one sperm fuses with the egg cell at the egg apparatus, whereas another sperm fuses with the polar nuclei to form triploid PEN. This diploid zygote, it undergo division to form an embryo, and this triploid primary endosperm nuclei undergo mitotic division to form a large endosperm, which is a nutritive tissue which will give nourishment to the developing embryo. In this slide, we can see the whole process of double fertilization or the life cycle of a flowering plant, where this is a flower, the calyx and corolla are accessory whorls, and rhizium and gynoecium are necessary whorls. In the androecium, there is a anther by the phenomenon of microsporogenesis, which is a reductional division. There is a formation of 4 haploid spores called microspores, which are otherwise known as pollen grain. This pollen grain, which undergo microgametogenesis to form 2 male gametes, which are otherwise known as sperms. Meanwhile, in the female structure, sex organ, that is the gynoecium, it undergo division to form megaspores, 4 megaspores. This phenomenon is called megasporogenesis. Out of 4 megaspore, 3 will degenerate and only 1 will remain as functional megaspore. This functional megaspore, by megagametogenesis, it is going to form 8 nucleated embryo sac. Out of 8 nucleus, 3 will arrange at the micropylar end, that is known as egg apparatus, whereas another 3 nuclei will remain at the chelagel end. They are known as antipodals, and in the center of the embryo sac there is 2 polar nuclei, where 1 nuclei come from the micropylar end and another nuclei come from the chelagel end. At the time of double fertilization, when the pollen grain come and fall on the surface of the stigma, they will start germinate and produce a pollen tube. Pollen tube grows through the stylar region and it reaches the ovule. When it reaches the ovule, it discharges the 2 male gametes or sperm into the embryo sac, where one of the male gamete fuses with the egg cell to form a diploid zygote, and another female 2 polar nuclei fuses with the male nucleus to form triploid primary endosperm nucleus. So after double fertilization, diploid zygote develops into embryo, whereas triploid PEN develops into endosperm. So endosperm is a nutritive tissue which will give nourishment for the developing embryo. Embryo later develops into a plantlet. Next, I'll move on to the significance of double fertilization. The two important products are obtained as a result of double fertilization. One is an embryo and another one is an endosperm. In double fertilization, the fusion of male and female haploid gametes results in diploid zygote formation, which later develops into an embryo, giving rise to a new plant. Other male gamete fuses with the two polar nuclei, give rise to an endosperm, which provide nourishment for the developing embryo. It utilizes both the male gametes produced by the pollen grain. There are chances of polyembryony and hence the plants have better chances of survival. It also increases the viability of angiosperm seeds. These are some of the significance of double fertilization. Now I'll come for post-fertilization changes in angiospermic flower. Once the process of fertilization gets completed, there are many changes takes place in the structure of a flower, which are as follows. The ovary consequently becomes the fruit, whereas the ovule becomes the seed, the ovary wall becomes the fruit wall, and the integuments of the ovule becomes the seed coat of the seed, where outer integument will become outer seed coat called testa, and inner integument will become inner seed coat called tegma, and the funicle get converted into a seed stalk. Diploid zygote, which is a product of fusion of one of the male gamete with the egg nucleus, it get converted into an embryo, and triploid PEN, primary endosperm nucleus, which is a product of fusion of two polar nuclei with one of the male gamete, results into formation of endosperm. And the nucellus, synergids and antipodals will degenerate. The sepals of the calyx and petals of the corolla will fall off. These are some of the post-fertilization changes taking place after double fertilization. In this slide, we can observe the post-fertilization changes taking place in the flower, where the calyx and corolla will wither off or fall off. Thus, ovary is converted into fruit, ovules are converted into seeds, seeds after liberation they will germinate to form one more new plant. With this, I complete the phenomenon of double fertilization in angiospermic plants. Thank you.